Hello everybody, I'm Silas, and welcome to Psilocybology. Let's expand our mind. This is a series where we talk about the research on psilocybin, the psychedelic prodrug found in magic mushrooms. We'll be breaking down and discussing the scientific literature, one study at a time, from past to present. If you'd like to support the show, please make sure to rate, review, and subscribe to the podcast. A quick announcement. This is the last week to fill out the January 2019 biannual listener survey, where you can provide feedback for the show. During the next episode, I'll announce what organization you all selected to receive the $100 donation. On that, for those who may not have been sure why you were asked to rank the organizations you'd like to see get the donation, rather than just voting for a single one, it's because I plan to use instant runoff voting. Instant runoff voting is a form of preferential voting where ballots are initially counted for each voter's top choice. If no top choice option has a majority of votes, then the option with the fewest votes is eliminated, and the votes that went to that option originally are redistributed based on the second choice listed on those particular ballots. That process is then iterative until one of the options holds the majority of votes. But anyway, on to today's episode. Today we'll be discussing the article titled The In Vitro Inhibitory Effect of Psilocybin and Related Compounds on Human Cholinesterases, published in 1963 by a collaboration between the University of Pittsburgh School of Medicine and Mercy Hospital. First up, cholinesterases are enzymes that break down the neurotransmitter acetylcholine. Acetylcholine is probably best known for its function at the neuromuscular junction, where motor neurons come into contact with muscle fibers. In this region, acetylcholine has the vital role of activating muscle contractions. Acetylcholine also plays a role in both the sympathetic and parasympathetic nervous systems. These are the systems in our body that are responsible for the fight or flight and our rest and digest responses, respectively. Finally, acetylcholine is found in some brain regions, particularly the basal forebrain, which is involved in arousal, attention, memory, and motivation. In addition to breaking down acetylcholine, it's important to know for this study that cholinesterases also break down butyrylcholine as well as methacholine. Now, both butyrylcholine and methacholine are synthetic drugs that act similarly to acetylcholine. Butyrylcholine is primarily used in a laboratory setting in order to distinguish between the two types of cholinesterases. The two types of cholinesterases have aptly been named acetylcholinesterase and butyrylcholinesterase. The reason they're named as such is because acetylcholinesterase has been shown to break down acetylcholine at a faster rate than it breaks down butyrylcholine, while butyrylcholinesterase breaks down butyrylcholine at a faster rate than it breaks down acetylcholine. Both cholinesterases break down both acetylcholine and butyrylcholine. It is just a matter of which one they have a preference for. The other key difference between these two cholinesterases is that acetylcholinesterase is primarily found in neuromuscular junctions and chemical synapses, and butyrylcholinesterase is primarily found in blood plasma. That said, research has found that in the brain, both of these cholinesterases can be found. This will be important to remember for later in the episode. Anyway, as for methacholine, also known as acetyl beta-methacholine, this synthetic drug can be used as a tool to detect the presence of asthma and chronic obstructive pulmonary disease. The only thing you really need to know about methacholine to understand this study is that it's broken down by acetylcholinesterase and not butyrylcholinesterase. So as a reminder, while acetylcholine is broken down by both cholinesterases, just at a faster rate by acetylcholinesterase, methacholine is just broken down by acetylcholinesterase. This will also be an important point to remember for later in the episode. The last thing I want to mention before moving on is that when conducting research into enzymes, the chemical that is being broken down by the enzyme is called the substrate. So as an example, if we were studying how acetylcholinesterase breaks down acetylcholine, then acetylcholine is the substrate. In this particular study, they used 3 substrates: acetylcholine, butyrylcholine, and methacholine. And again, as another reminder, acetylcholine is found naturally in the body, while butyrylcholine and methacholine are both synthetic. However, both acetylcholinesterase and butyrylcholinesterase are found in the body. Okay, so now that we have that foundation, let's talk about why the researchers were interested in how drugs like psilocybin may inhibit cholinesterases in the first place. In their introduction, the authors discuss how research into LSD found that LSD administration appeared to cause an increase in acetylcholine levels in the brain. Subsequent follow-up studies into LSD observed that LSD appeared to inhibit plasma cholinesterase activity. I'll note that since butyrylcholinesterase is primarily found in blood plasma, usually whenever you hear plasma cholinesterase, it is referring specifically to butyrylcholinesterase. Given this prior relationship and the interest during the 60s for determining how hallucinogens work at a neurochemical level, the researchers primarily wanted to examine 3 things. First, does psilocybin, like LSD, appear to inhibit butyrylcholinesterase activity? Second, does psilocybin also appear to inhibit acetylcholinesterase activity? Third, how does the inhibition of cholinesterase activity in response to psilocybin compare to that of other hallucinogenic drugs, in this case bufotenine, and non-hallucinogenic drugs, in this case serotonin? According to the researchers, since butyrylcholinesterase is primarily in the blood plasma, its inhibition by psilocybin, while interesting, likely wouldn't explain the psychoactive properties of the drug, since acetylcholinesterase is the primary cholinesterase of the brain. If instead psilocybin did appear to also inhibit acetylcholinesterase, then the authors hypothesized that this inhibition is in fact contributing to the psychoactive effects of the drug. The reason bufotenine and serotonin were selected as comparators is because, as we discussed previously on this podcast, all 3 drugs are structurally very similar. The design of the study at a high level is rather quite simple, but it gets a bit more complicated when discussing the specific tests they conducted. At the high level, the researchers examined how psilocybin, bufotenine, and serotonin inhibited acetylcholinesterase and butyrylcholinesterase by administering these drugs to solutions that contain the 2 enzymes. Taking the study step by step, first the researchers took 3 groups of human tissue samples. One that was comprised of blood plasma, one that was comprised of red blood cells, and one that was comprised of gray matter that was taken from recently deceased hospital patients. Now remember, the samples with blood plasma are assessing butyrylcholinesterase. The samples with red blood cells are assessing acetylcholinesterase. The samples with gray matter, although they primarily contain acetylcholinesterase, are assessing both butyrylcholinesterase and acetylcholinesterase. We'll talk about how they distinguish the 2 in a moment. So once the researchers had these 3 sets of tissue samples, the next step was to further divide up the 3 sets of samples so that we'll end up with a total of 9 sets of solutions. For each tissue type, 1 set of solutions will be mixed with psilocybin, another with bufotenine, and another with serotonin. The final step, after all the sets of solutions have been mixed, is to add the substrates. The reason I keep saying sets of solutions, plural, is because the researchers ran their tests using 6 different concentrations of each of our 3 drugs of interest, and also conducted the entire set of experiments twice. The reason they used varying concentrations of the 3 drugs has to do with how they decided to measure inhibition. To measure inhibition, they assess what's called the half maximal inhibitory concentration, or IC50. This is a quantitative measure that indicates how much, i.e. the concentration, of a drug is needed in order to induce a 50% inhibition of a given biological process. Stated differently, the IC50 measures how much of a drug is needed in order to make one of the enzymes 50% as effective at breaking down one of the substrates. A key piece of the puzzle, so to speak, that I had to think about when reading the study is that all three drugs to some extent inhibit both of the enzymes. The aim of the study wasn't just to detect whether there was or was not inhibition caused by psilocybin, but rather to detect if there was any clinically meaningful inhibition caused by psilocybin. What I mean is the inhibition caused by psilocybin meaningful in that it is what's causing the hallucinations that are produced by psilocybin? That is why they wanted to compare the inhibition caused by psilocybin to the inhibition caused by bufotenine, another hallucinogenic drug, and serotonin, a non-hallucinogenic drug. If psilocybin caused no greater inhibition than did serotonin, this would indicate to the researchers that this inhibition isn't what's responsible for the hallucinogenic effects of psilocybin. And if psilocybin did in fact produce greater inhibition than serotonin, and the hypothesis is that that's what's causing the hallucinations, then we should also see similar inhibition caused by bufotenine, since it produces hallucinations as well. Before moving into the results, we need to take one quick step back and discuss the substrates and solutions again. For the plasma tissue, both acetylcholine and butyrylcholine were mixed into the solutions. Since blood plasma consists of butyrylcholinesterase, then any inhibition in the breakdown of these two substrates indicates inhibition of butyrylcholinesterase. I will say one limitation to the study is that the researchers assumed acetylcholinesterase is not present in blood plasma, and it looks like research has since found that there is in fact some amount of both cholinesterases in blood plasma. For the red blood cell tissue, the researchers added both acetylcholine and methacholine, since this sample consists of acetylcholinesterase. Any inhibition in the breakdown of these two substrates indicates inhibition of acetylcholinesterase. For gray matter tissue, this is where it's important to remember that both acetylcholinesterase and butyrylcholinesterase are found. For this set of solutions, the researchers added methacholine and butyrylcholine. If we also remember back to when we discussed methacholine is only broken down by acetylcholinesterase, and butyrylcholine is primarily broken down by butyrylcholinesterase. So, any inhibition in the breakdown of methacholine indicates inhibition of acetylcholinesterase, while any inhibition in the breakdown of butyrylcholine indicates the inhibition of butyrylcholinesterase. So finally, after all of that, we're ready to get into the results. If you can believe it, this entire article is only 2 pages. Anyway, starting with the sets of solutions containing blood plasma, the researchers found that both psilocybin and bufotenine had a greater inhibitory impact on the breakdown of acetylcholine and butyrylcholine compared to serotonin. This was interpreted to mean that both psilocybin and bufotenine inhibited butyrylcholinesterase to a greater extent than serotonin. For the red blood cell tissue samples, the researchers found no difference in the breakdown of acetylcholine or methacholine across the groups with psilocybin, bufotenine, and serotonin present. This was interpreted to mean that neither psilocybin nor bufotenine inhibited acetylcholinesterase to a greater extent than serotonin. And lastly, for the gray matter tissue samples, they found that psilocybin and bufotenine appeared to inhibit the breakdown of butyrylcholine to a greater extent than serotonin. However, there is no significant differences in the breakdown of methacholine across the 3 drug groups. This was interpreted to mean that, consistent with the earlier 2 findings, psilocybin and bufotenine inhibited butyrylcholinesterase, but not acetylcholinesterase at a greater extent than serotonin. Ultimately, based on these findings, the researchers conclude that they do not believe the inhibition of cholinesterases by psilocybin is what's producing the hallucinatory effect of psilocybin, since it does not appear to greatly inhibit acetylcholinesterase, the primary cholinesterase in the brain, but rather butyrylcholinesterase, the primary cholinesterase in blood plasma. Although there are some other limitations to this study, I'd say that largely, the biggest, is that since the study was done, researchers have found that selective butyrylcholinesterase inhibition does lead to elevated brain levels of acetylcholine. So their conclusion, that because psilocybin appears to selectively inhibit butyrylcholinesterase, that acetylcholine must then not contribute to the hallucinations caused by psilocybin, is ultimately unfounded based on these specific results. I'll actually link to the study that found elevated acetylcholine after butyrylcholinesterase inhibition in the episode transcript. It's a really interesting paper that's actually focused on the implications for Alzheimer's. Interestingly, and this is mere provocative conjecture, but if their paper is right, in that inhibition of butyrylcholinesterase may reverse the course of Alzheimer's, then psilocybin might actually prove to be a potential treatment option for Alzheimer's, given its inhibition of butyrylcholinesterase. I firmly believe that psilocybin is a transdiagnostic medicine, and my belief in that is reaffirmed the more I keep learning about it. Before we end the episode, I also want to place the study into a bit of a bigger picture as far as drug research at the time goes. This study can be thought of, in part, as aimed to help distinguish between three types of hallucinogens: psychedelics, dissociatives, and deliriants, all of which were drugs of immense interest during this period of time. While psychedelics include the drugs like psilocybin, bufotenine, LSD, and mescaline, dissociatives include drugs like ketamine and PCP, and deliriants include drugs like JB329, also known as Ditran, which we talked about on episode 6. One of the key physiological features of deliriants is that they are anticholinergic, meaning that they block the action of acetylcholine. Given how important acetylcholine is for that group of hallucinogens, researchers wanted to see if acetylcholine also played a key role in the function of other types of hallucinogens. In this regard, the study has some additional value in showing that while deliriants lower acetylcholine activity, psychedelics appear to increase acetylcholine activity, since inhibiting cholinesterases would mean less acetylcholine is broken down, and thus more activity. This helps to explain why people don't experience delirium while on psychedelics. So with that, that's the end of our 20th episode of the podcast. I actually can't believe the podcast has already been going for 5 months. I'm incredibly excited to see where it keeps going through 2019. As always, I really hope you enjoyed the show. If you have any questions, feedback, or ideas for the podcast, please let me know. You can find out all the ways to reach me on the website, psilocybology.org, where you can also find the full transcript for each episode if interested. Thank you all for listening. I'll talk to you next time.